So let's start. It's a great pleasure to introduce Melanie Fernandez-Pradier. She's I was trying to roll my R more, but there you go. Pradier. She's currently a postdoctoral fellow at Harvard University, advised by Professor Doshi Velez. She works on interpretable machine learning, probabilistic models, and biomedical applications. No wonder she's here. She's a member of the Harvard Data Science Initiative and CRCS Institute. She received her PhD on Bayesian on parametric models for data exploration at the Universidad Carlos III Sciences. Is that how you pronounce it?
Carlos III, yeah.
Carlos III. Oh, yeah. I was trying to show off there. In Madrid in 2017. In 2015, she was a research visitor at the Memorial Sloan Kettering Cancer Center in New York. And before that, she spent 2 years at Sony U Research in Stuttgart and Sony Corporation R&D in Tokyo. As a side project, she also is a user advocacy advisor for the Eon Case tech startup. And she's interested in Bayesian neural networks, hierarchical representation, approximate inference, dependent random measures, and personalized mental care. All right, let's start.
All right. Thank you, Nicola, for the introduction. Is the sound good? Okay, yes. I'm very much excited to be here and share my work with all of you. So yeah, I'm a postdoc at Harvard, an active member of the Harvard Data Science Initiative. So I develop in my work machine learning methodology to solve impactful problems in healthcare and medicine. And regularly, I work in very multidisciplinary teams. So, for example, I collaborate with psychiatrists in Massachusetts General Hospital currently, also with experts in human-computer interaction and biostatisticians. And in the past, I've worked with oncologists, with economists, sports scientists, etc. I care very much about building models and developing inference in a way that we get the right properties for the right tasks. So today I'm going to talk about how to incorporate expert knowledge into the model design. So before starting, I would like to thank my collaborators. So all this work would not be possible with all of them. And all right, as you know, we are living a machine learning revolution, right? Machine learning systems are everywhere. So we have recommendation systems, automatic translations, self-driven cars are coming. And we have never been better at distinguishing cats from dogs. So this is fantastic, right? But jokes aside, it's true. So if you Google machine learning, you get 3 billion entries. Interestingly, if you Google Donald Trump, you get half of that. So machine learning is really there, right? It's amazing. And you look at papers and the press, machine learning is revolutionizing our life with breakthroughs in natural language processing, with in biology, in healthcare. We are very good at handling images. And recently, so these 2 pieces of news are from this week. This is from yesterday, and 2 days ago. And this one, so it's from exactly 1 week ago, and we can see how machine learning systems are getting used to discover and design new drugs in the lab. That's very exciting. However, if we look at other types of articles, we also see some mistrust. We also see some issues there, right? So AI systems are also very easy to fool. So people are talking about these adversarial examples. Then we have sometimes bias. We have a need for fairness. Then we also, so we have a problem, which is that we talk about big data all the time, but in fact, even in some settings, it's very hard to actually get enough data of the right instances. For example, in healthcare, patients are unique. And even if you gather tons of data, which is sometimes not even possible because of privacy issues, you still will get unique profiles of patients. So we need methods that are able to deal with this so-called small data within big data. That's a need. And generally speaking, so we need methods, models that are easy to interpret, such that we can actually diagnose and understand whenever all these issues come up. So in my work, I care very much about interpretability. That's the ability to present in understandable terms to a human. So given a machine learning system and a domain expert, I need to provide some explanations through interpretability. This has become very important, especially in the European Union since 2018, given the General Data Protection Regulation. So according to this regulation, now every time that an algorithm makes a prediction, the user has the right to ask for an explanation. Why did I get that prediction, right? And because of that, there is a huge pressure both in industry and academia to work in developing these explanations. So that's one side of the coin. The other side of the coin, and I will argue it's much less talked about, is the so-called expressivity, which is we need tools, we need statistical methods that allow us to encode the right assumptions and desiderata into the model. Will be from the domain expert to the machine learning system, right? And so this completes a very beautiful loop in order to push for breakthroughs within science. So this is central in my research, and now I would like to give you an overview of my research journey. So I actually started working in information theory just at the beginning of my PhD. And then I switched off into machine learning, specifically in the Bayesian modeling framework. And I work in a variety of subdisciplines in collaboration with experts and trying to bring this both interpretability and expressivity in each domain. So let me give you a little bit of an overview. So first, I work in sports sciences. So here the question was, how does aging actually impact our athletic performance? So we had data from marathon, and here the goal was to model the finishing time such that we could compare the running time among athletes, regardless of their age and gender. So it was a fair system, and that could be used to decide who should enter as an entry requirement to the marathon or to reward runners. So this was one project. This is the marathon, the New York Marathon profile, and we found clusters of how people actually run. That was very interesting to see. I've also worked then on economics. So the question here is which factors make countries wealthier than others? Ultimately, we want to answer this. So we look at trade data. So we had countries times products. And the idea here was that if we look at what countries export, we could maybe have an idea of what are the latent capabilities of those countries. And look at, so through these latent capabilities, we could actually get some indicatives of country development and see how these capabilities evolve through time. Then another domain I've worked on, it's cancer research. So this, I've worked in collaboration with the Memorial Sloan Kettering Cancer Center. And I did genetic association studies, trying to understand what are the underlying mechanisms of cancer. And so this is currently submitted to Nature Communications. And I'm going to talk today in this more in detail about this other project in collaboration with Roche, which tries to look at biomarker discovery. Then finally, and more recently, now here at Harvard, I work on personalizing antidepressant prescription. So we look at electronic health records and try to predict when a patient is going to be stable or when a patient is going to drop out from their treatment after having been given a certain antidepressant. And so 2 of these publications have or just been accepted last week to the JAMA Open and the Nature Translational Psychiatry. So this is exciting. Oh, yes. And obviously, so all these domains need a very important component from machine learning methodology. And I would say I work in these two categories of model design, so trying to design expressive priors to encode these assumptions. And as well, I also work on inference, so trying to make inference better, more precise, and so scalable for whatever down task application we have at hand. Yeah, so I'm very happy to talk about all these projects later in the questions, so that's why I wanted to give an overview. And now, so this is my roadmap. Today I'm going to talk about this. So first I will start with the cancer research project in collaboration with Roche. Any questions so far before I jump into the first project? Okay, awesome. So let's go into our first quest, which is biomarker discovery. Here I'm going to talk, I'm going to use a loose definition of biomarker, which is any variable that can be used as an indicator of a particular disease state. And this project is directly inspired by a real need. It's an application that so we have a phase 2 clinical trial data for a particular immunotherapy treatment for liver cancer. So Roche had this data, and the way it works, so we have here the cancerogenous cell, and this cell has some markers, and the idea was if you administer some antibody to this patient, this so-called CD16, so it's going to bind to those markers that are already present in the cancerogenous cell, and it's going to make it easy for the immune system to actually find that cell. So these are the NK cells that are going to come and kill the cell, and you're saying, hey, this is a bad cell, just come and kill it. That's the way the drug is meant to work. And this is a phase 2 clinical trial, so we are trying to assess what's the effectiveness of the treatment. So there has been a previous study from 2016 from Abu Dalfa, the team at Roche, and they run classical statistical tests. The conclusion of this study was that there is no evidence for the treatment effectiveness. So, well, that happens, right? So we create the data, we try to see whether it was effective or not, and guess what? It was not as effective as we thought. Well, where we are going now is so they gave me this data, and the goal was to actually try to see, okay, is there actually a subgroup of patients for which this drug is actually working, instead of just looking at the effectiveness across the whole population? Can we actually target and get a subgroup that it will actually benefit from the drug? So the answer, and where we are going, is that using a novel Bayesian approach, our conclusion was that indeed we can find some patients for which the treatment is effective. So this was very exciting news to actually confirm some of the intuition of the clinician, which was that, hey, we have this data, we think there is something there, even if we haven't found it yet, and yeah, so can you do that? Can you do something about that? And let me tell you why this problem is hard, right? Why didn't they find it before? Well, so the first challenge, as you can imagine, is that biology is complicated. We have very complex correlations between the biomarkers. We will have to deal with that. The next challenge is that patients are very heterogeneous, as I was saying. And not only that, we have very few data. This study has less than 200 people. 200 patients, and we cannot gather more just because of ethical reasons. So that's what we have. And then another challenge, so if you have, for those that work with real data is a mess, right? They're missing, and also they have data types that are very different. So we have continuous, we have discrete, ordinal, categorical, so each feature is just very different from the next one. And that makes it very hard for modeling. and often what people will do is just assume a general model, simplified for all the data types, and this is sometimes a very relaxed assumption. So we will have to consider that. And finally, and most important, is the fact that we have to be able to distinguish the drug effect from the natural response of the drug. And this is hard just because we only get to observe the response of patients once. Either you get the treatment or not. And how can I actually distinguish whether it was a natural progression or because of the drug? All right, so those are our challenges. Now, in the following slides, I'm going to tackle each one of these and show you how we actually build our model to get that. So the first challenge is how to deal with complex correlations. And for that, we resort to a model called latent feature model. So we are doing basically a probabilistic matrix factorization here. So given our data, n patients, and the observed features, the observations, we are finding this matrix factorization. So any data point here, say, for example, we have this column is glucose in blood. And so we have, for this particular patient, 173 milliliters glucose. What we are doing is finding a linear combination of terms to explain this observation. So each position here is going to be the multiplication of this row, one row by one column. And you can think of this matrix B as a dictionary matrix. So each row is going to capture a correlation pattern of how the different biomarkers, how the different observed features actually correlate to each other. So this is the dictionary matrix, and the first matrix is capturing how much of these features gets activated for each specific patient. So one row tells me how much I need, how much is active for each of these correlation patterns. So that's the idea. Now, let's go into the challenge number 2, which is, these patients are very heterogeneous. So ideally, we would like to have a model that is able to cluster, in a way, the patients that are similar to each other, right? And we're going to do that with latent features that are binary. So if you make this matrix binary, what it turns out you can interpret the values as saying, okay, this particular patient has the second and the fourth features, so the correlation patterns, active. So it's highly interpretable, and if you do that, then it's actually easy to cluster patients together. So I'm going to be clustered with all the patients that have the same binary signature, the same vector than me, because they have the same active correlation profiles, if that makes sense. Okay. And I remember we have very little data, right? So how can we deal with that? So here we go into the Bayesian non-parametrics framework. So Bayesian is very useful. It's a very useful framework to handle uncertainty, to reason about the noise. And so what we do is we are able to actually compute a posterior distribution of the parameters given the data. And this is going to be proportional to a likelihood term times a prior over the parameters. So, okay, which parameter here we care about is actually the number here of latent features. Like, how many correlation patterns do I expect to see in my data? Well, a priori, we have no idea, right? Ideally, we would like our model to learn this K automatically. And this is the advantage that we get from using the non-parametric part. So we are able to adapt model complexity depending on how much data we get. And so you can think of it, if you have certain observations, certain patient information, you might get 4 clusters. Now, if you get more data, then the model is able to learn, hey, I need more clusters. So this K, this number of latent features K, is able to adapt automatically. So we use this framework, and specifically, we use a prior based on the Indian buffet process. This is a Bayesian non-parametric model, a prior for latent feature models. And the way you can think of it as, so in the same way that if you sample from a Gaussian, you get a real number, right? If you sample from an Indian buffet process, what you get is a binary matrix with a varying number of columns, right? And this is exactly what we want. We want to be able to learn a binary matrix, but whose number of columns is unknown. And that will be also learned from the data. So we denote this prior like this. Z is a draw from the Indian buffet process with parameter alpha. And you can think of this parameter, called the concentration parameter, as a meta parameter or hyperparameter that tells you a priori how I expect my number of latent features to grow with data. Okay, so let me give you one more challenge, which is how can I deal with different data types? So remember, here each column is very different. Discrete data, positive, then real value. We have a mess of data, and so people often will just assume, okay, everything is Gaussian distributed, and this is a very strong assumption. What we do here is to assume a link function specific for each data type. So this is just a cartoon. So in fact, we're going to have a link function for each observed feature D, or for each column, and that will depend on the specific type of the data of that column. So what you get, if you do that, the advantage is that you can think of, so this multiplication here, you can treat it in the same space for all the variable for all the data types, say in real space. And then you have a T transformation link function to map to whatever the observational space was. So if you had ordinal data, then this T will make sure that the matrix factorization real value here that you gets mapped to the ordinal data. Okay, so now let's put all these elements together, and this brings me to the first contribution I would like to mention, and this is the general latent feature model. So everything I say here in cartoons can be formalized within this probabilistic graphical models methodology. And so in this work, we present a package, open source package, that is able to discover these complex correlations in data. And it can deal with data heterogeneity, as I was saying, in the rows. So either patients or whatever data you have. Noisy observations, missing data, because we're dealing with uncertainty in a Bayesian manner, as well as different data types. So if you have a data set that is highly heterogeneous in the columns and the different data types, hopefully this is a useful model for to try. Also, and very convenient because it adjusts the model complexity. So you don't have to set or try different number of latent features. We have implemented a wrapper in Python, MATLAB, and R to make it more accessible to researchers. Okay, so yes?
What's phi d on the...
Yes.
The parameters of the link?
Phi d are the parameters of the link function, yes.
Are you inferring the link function too, or plugging it in?
No, so the link function is fixed. However, we have a set of link functions, and in the package we have a preprocessing step where the data gets normalized and set up in a range such that the hyperparameters of this link function will actually be appropriate for that. Yeah. Yes.
Discuss the binarization process a little. So how, if you have a continuous variable, for instance, how does that turn into the 0 or 1, or if it's categorical or...
Oh, right. So the binary part comes directly from assuming the Indian buffet process prior. So there is no binarization process. So we just said, okay, these variables here are going to be discrete, random variables. And and so the Indian buffet process, so whenever we do inference, we are actually inferring the probability distribution of being 1 or 0. Yeah. Any other question? Okay. Okay, so this is the first contribution, and now let me recap where we are. So I talk about these 4 challenges. So we are dealing with complex correlation by doing this probabilistic matrix factorization or finding latent features. We deal with patient heterogeneity and by the fact that the binary is binary, and the few data is actually dealt with the Bayesian framework and Bayesian. So the non-parametric part allows us to actually learn the model complexity, and then the data types are dealt with link functions. Now, how about the last challenge, the distinguishing between drug effect versus natural response? So this is a very important question in healthcare in general. The way we tackle this is to actually design the prior in an expressive way by allowing the model to learn treatment-specific latent features. So now these additional latent features can only activate for those patients that are in the treatment arm. So here we will always have zeros for those that are in placebo. And why is this useful? Well, you can think of it also, so when we do the matrix factorization, it's like we are finding a basis, right, to explain patient sickness in general, right? And then, so once we have that space, we are trying to see, okay, do I need additional dimensions, additional bases to actually explain the differences between patients in the placebo arm and the patients in the treatment? So the benefit, and this brings us a very interpretable output, which is that whatever I capture in this so in these extra rows, these correlation patterns are going to be linked to whatever the treatment the treatment effect is. Yeah. Yeah. Oh, yeah, because I was saying that here in this matrix, every row can be seen as this correlation profile of how the biomarkers correlate to each other.
Right, but it's per patient. You're not adding patients. You're adding.
We're not adding patient. No, I'm talking about the latent space. Yes. Or another way to look at it is here. So the patients. So remember, there is one row here for each patient. So I'm patient with first, second feature active. Okay, and these are the general latent features. And then for the treatment specific latent features, that might be active or not for me, yeah.
Not that's adding a column, but maybe I don't understand the matrix.
No, so, yeah, sorry. So here, we are adding a column to the first matrix, this here. Yeah. And that means we are also adding rows to the second matrix, which is a dictionary, because we get additional profiles for the treatment.
The extra rows, not extra patients, it's an extra set.
Oh, no, they're not extra. So, yeah.
Extra set of correlations.
Exactly, yeah. Exactly. So this matrix is a k number of latent features times the observed features. Yeah, no, please. Yeah, any question is super welcome. I like when it's interactive. So, okay, so now we can formalize and enhance our graphical model. So this is actually easy to do by adding additional random variables into our graphical representation. And so this is the second contribution I would like to mention. So case control Indian buffet process. It was published in BMC Cancer last year. By the way, I didn't mention the other work was published in, is actually now going to be published in Journal of Machine Learning Research this year. Okay, and let me jump directly in the results of this model. What did we find? Okay, back to the real problem. So we have 180 patients here. 60 took the placebo and 120 took the drug. And the measure for success that we monitor is progression-free survival. So that's how much time it elapses until the tumor actually grows its size. And what we found, our model found 3 features. So remember that the model is actually learning the number of features that is needed. 2 of these features were generic, and 1 feature was treatment specific. This is why here, so it's going to activate only for placebo patients. And here, the rows correspond to the different binary signatures that the patients get. So these are, each row here is a subpopulation of patients that have the same binary signature of the latent features I learned. So the first 4 groups correspond to the patients that took placebo. And interestingly, you can see that the 4th subpopulation, so patients that have the first and the second latent feature active. So if we look at progression-free survival, our measure of interest, it actually gets 4 times more longest period until the tumor increases its size. So that means that patients that are in this category actually have a very good natural response to the disease. Okay. Yes. So I have. Oh, I don't have any slide. Yeah. So we monitor the whole distribution as well. Yeah, I don't remember from the top of my head, but it's in the paper. Yeah.
It's significant. You can still identify that the 718 is significant difference from the alpha.
It is. Yeah. It is significant. Yes. Yeah. So okay, and now let me talk about the second. So the patients in the treatment arm. And specifically, so if we look at the treatment-specific feature, so if you look, whenever this feature is active, the progression-free survival actually gets double, meaning that we have found a latent feature that is indicative. So whenever it gets activated, it's actually it's actually correlated with a much positive response to the drug, right? So this is very exciting news. Because now I can know for which patients this treatment is going to be more effective. So anytime the latent feature here is active, I expect the patient to be more responsive to the treatment. Okay, and now what we can do is actually see. So remember, the task is to do biomarker discovery. So we want to know which biomarkers are actually meaningful to either assess natural response or actual effectiveness of the treatment. So for that, we look at, in particular, the biomarkers associated to this feature, F3. And so I'm happy, I have extra material for this, so happy to chat that in the questions. We did statistical tests for the biomarkers associated to this latent feature. And interestingly, we found out that the statistically significant biomarkers were actually correlated to the mechanism of action of the drug. So what did we find out? Let me recap. First, our method is able to identify subgroups for which the treatment is especially effective, number 1. And number 2, we were able to give to the oncologist a list of relevant biomarkers, so with effect size and sign of the effect size, so biomarkers that indicate that the drug is actually acting as expected. Yes.
Can you go back 1 slide?
Yes.
Sorry, this might get involved, so stop if you need
No. That's fine.
So if I'm understanding correctly, there's 3 latent variables, F1, 2, 3.
Yeah.
I don't know how to interpret them. Maybe you do.
No,
Okay.
but
We can come back to that.
We'll come back to that.
But the top 4 rows are all patients who didn't get are clusters of patients who didn't get the drug. The bottom are patients
Yes,
who did.
that's correct.
And what you're seeing in the top 4 is that if a patient has F1-1 and F2-1, that there's something natural about that. They didn't get the drug, but something natural about that confers better prognosis.
Exactly.
Okay.
Yes.
Then in the bottom ones, same deal. If you've got 1 and 1 and F1 and F2, you're in good shape. But
Yeah.
now what you're saying is if you also have F3, what is F? Is F3 is not. It's some latent variable having nothing to do with getting the drug or not.
So F3 is a treatment-specific latent feature, meaning that it's a feature that can only activate if you have been given the treatment. Yes. So this is why here you can see.
Interpretation. That's I understand what that means. Like biologically, what does that mean? It's something that only. Is that what's coming next?
So biologically, it could like the reason why we are interpreting this feature as a positive, like positively indicating, hey, the treatment is working, just because we look at the progression-free survival on these different. So I mean, like generally speaking, for this method, what happens is you get extra, treatment-specific features that is grouping your patient in a different way.
So what's an example of a treatment-specific feature? Maybe you were about to tell me.
Yeah, no. So this is actually the correlation profile associated to this treatment-specific feature, meaning that if you have F3 active, then you are going to get higher levels of these so NK markers here, and you're going to get lower levels for this other biomarker. This is what it means. Yes.
Biologically, the assumption here is the natural killer cells won't do anything unless you boost it with the drug. Is that how I should interpret it?
Can you repeat again?
You're saying,
Yeah.
for example, the NK cells, that's a big high one, right? Okay, so the NK cells, the drug is supposed to boost that, but that might happen even if you didn't have the drug, right?
So, okay, yeah, it's a great question.
NK cells active even if you didn't have the drug, right? Or is that not correct? That's what I'm trying to understand.
Right. So the key point here is the fact that this latent feature will never. So this profile only activates with patients that have taken the treatment. So that means people in the placebo arm didn't get this profile. Otherwise, it would have been learned as a generic latent feature. But our model was able to learn. Okay, I need these, general latent features. This one only appears for those that do the treatment.
Because NK cells don't show up in those that don't have it, or because of something you're doing in the math differently?
Oh, it's because of how we have set the model in a structured way, like by just saying this feature cannot activate to placebo. That's the magic of.
True biological.
So.
Even if you had a placebo, that the NK cells would activate.
So what I'm saying is, okay, it's just I'm not sure I get your.
We can take it off.
Yeah. Okay.
Let me ask a related one.
Yeah.
Maybe an interpreted, like in general, all the features that are specific to the treatment group, they seem to correlate with an active immune system.
Yes.
So is an interpretation that if you have a more active immune system and you target cancer cells, like with treatment, then you kill more?
Yeah, exactly. That's definitely the.
A more active immune system without the drug, also.
You can, but if you're not targeting the right, like if you don't give the treatment that points the immune system what to target, then you just have more NK cells.
I see. And they won't do.
They just
the right
float
thing.
around.
Got it. Okay.
Yeah. No, and in fact, so we have these profiles also for feature 1 and 2, and there were also indicators of natural strong immune system for those patients by just looking at these latent features. So I guess the interpretability part, or like the meaning in a biological sense, would be by inspecting what are the correlation profiles of each of these latent features, and then work with oncologists, that's what we did, to actually give a meaning or say, okay, why these biomarkers correlate to each other. Yeah.
Are you showing that or not?
I have it in the extra slides. We can maybe talk. Yeah, so I want to make sure I get into the other project. So yeah, but thank you. This is very great questions. Okay, so let me now talk about a different project, which is more oriented into machine learning methodology, but it has very important implications in different predictive problems. So I'm going to talk about inherent limitation from state-of-the-art so neural network formulations. And so, as you know, neural networks can fit very complicated trends in data. So if you have an input X and an output Y, we are able to basically learn any function because of the flexibility of neural networks, right? They're very expressive. And this has led to very successful stories with images, with text, with sound, so speech. This is great, but now, what if the neural network is wrong, and this is a high-stakes situation, right? Like, what will you do? Well, it turns out, so imagine here we are in an ICU setting, right, and you are modeling blood pressure now. And now I'm telling you, okay, I'm using this neural network to actually assess whenever the blood pressure of the patient is high or low. And based on that, I'm going to decide if I send a nurse or not to the patient, right? Yeah, and neural networks sometimes overfit. So how do we feel about that? That's, instead of giving a point prediction, what we might want to get out of the model is actually not only that, a point prediction, but also an interval, right? Some uncertainty around my prediction. So for a certain, for example, time here, I would get not only one value, okay, 100 beat per minute heart rate, but also plus minus 20 minus whatever. So this is the blue area here, and I claim that uncertainty estimation becomes crucial whenever we are in high state settings, either healthcare, climate change, stock markets, etc. So this is great, but sometimes in these high stake decisions, we also have functional, so we have prior knowledge. For example, we already know that heart rate at rest is between 60 and 100 beats per minute, right? So if a neural network tells me, hey, the heart rate here is minus 10, well, I know I don't have to believe that. We might also know a priori how the variation of air pollutants should vary, either slow, fast, or how much volatility there is in a certain stock market. This is information that domain experts have already. And if we know that, the key point is that all these assumptions can actually translate into restrictions of the learned function. So that this is going to shape the types of uncertainty that we'll be able to get, right? And so now, within the neural network framework, how can we incorporate such desiderata into the model? And let me ask you, so do you know any model? Do you know how an easy way to incorporate some notions of this is amplitude variance, so range, like where my function should actually move or vary, or notions of length scale, notions of how fast or slow, how wiggliness our function should be? Well, it's not an easy task with neural networks. But there is another model, and this is a Bayesian nonparametric model called Gaussian process that actually help us to incorporate that prior information very easily. And the way it does, so there are two parameters that allows to actually model, so the wiggliness of the function, this is controlled by this hyperparameter, the length scale. So what we say a priori is, hey, I expect my function to vary very fast. And because I say that to the model, then in regions of the space where I have no data, I will be more uncertain because I know that my function can vary very fast. So I'm going to reach potentially, bigger regions. And then the other parameter is the amplitude variance that controls what's the range of my function, in which range I expect my function to move. So interestingly, we also have the concept of stationarity. Stationarity means that these properties are so stay the same in the whole input regions. And that you can think of it as I want my uncertainty to be I want to be as equally uncertain if I have the same lack of information in the whole input space. I'm going to come back to this notion of stationarity later on, but just I want to point out that Gaussian processes are able to incorporate stationarity. Okay. So Gaussian processes are great, and so that's it, we solve it. So what we can do is just convince everybody to give up on neural networks and just use Gaussian processes, and that's it. But it might be hard, people are going to stick with neural networks, not only because they're widely used and there are many tools available, but also they have advantages. No? They have a parametric expression. I can compute the y's with a closed form analytical expression from the x's, and that has benefits if you want to do later on extra computation, compute the gradients, or do Bayesian optimization, and so on. And also, they are very fast at both training and evaluation. So if you want to deploy some mobile application, evaluation time is crucial for those applications. So what's the research question in this project? What I want is to develop a Bayesian prior for neural networks that is able to encode certain properties like a GP, but still is a neural network. That's our goal. And we want not only to be able to encode the stationarity properties, but also to be able to specify these two basic properties so the notions of length scale and amplitude variance in a decoupled fashion, such that the user can say, hey, if I know my range of heart rate, I can specify the amplitude variance. And if I know the variation of my function, I will specify the length scale. That's our objective. Any questions so far? Okay. Awesome. So let me just briefly introduce you what the current Bayesian framework is for neural networks. So Bayesian neural network is a Bayesian prior where you're actually assuming independent IID Gaussians on each network parameter. And the way it works, so we're putting a prior over the network parameters, and this implies automatically a prior in function space. So if we let's look here at the prior. These are samples from our prior. So if you have one set of parameters, you get a function, right? So if you sample multiple network parameters, you get a set of different functions. So these are the functions that my model can represent even before getting to see any data. Okay? And then when we get to see actual data, then we need all these functions to actually fit, to actually go through the data. And still we get a probability distribution in my function space through putting a prior over the network parameters. And now the question comes, so remember we want interpretability, expressivity, so what does a prior over weights mean in function space? So, because what I care about is to say, I expect my function to be in a certain way. I expect some properties of the function. And now what should I put here on the network parameters in order to get those properties, right? So that question is not easy. And in fact, not only it's hard, but I claim that certain properties are just impossible to get with state-of-the-art neural networks, okay? And you might have noticed already in this slide, it's like, hey, so here I have seen zero data, and already I get functions, I get more uncertainty or more variance, amplitude variance, around the origin. So I haven't seen any data, and I'm already being more uncertain in certain regions of the space than others. So why would I have this type of asymmetry in my uncertainty if the data is not even there? That's very disturbing if you think about it. So in fact, so here we see that affects the uncertainty I get in my posterior. So here we have a toy illustrative example of 4 data points. Okay, and so here we have the mean predictive function, which is more or less a line, and we get the green region is the uncertainty that I've learned with my model, and as you can see, we have much more uncertainty in the center, in the around the origin, than on the sides. And yet, these points are equally spaced, what I would ideally expect is to have equal uncertainty. I should be equally as skeptical of my predictions within the whole region, right? Now, the question is, can we design a novel Bayesian neural network, a Bayesian prior for neural networks that is able to exhibit stationarity? So the answer is yes. That's what we do in this project. And yeah, so we get a behavior similar to a Gaussian process here. Okay, and so our model builds upon this family of neural networks. So could you maybe raise your hand if you have heard about radial basis function networks? Anybody? Yeah, 1, 2, so yeah. So not many people, this is actually a very old model. It's from the 80s. But for some reason, it's not very widely known in the machine learning community. It is very much used in geospatial applications, engineering, materials, and it has gained renewed attention lately with some optimization papers from Zubris Fra and folks here at Tübingen. So what's this model? So I will just give you an intuition. It's an alternative parameterization of a neural network. So it is a neural network, but instead of having weights and biases, we're going to have centers of the activation units and scales. So centers tells me where in space I'm placing the activation units, and scales tell me how wide, how spread is the influence of the activation unit. And so there is, mathematically, if you look at this expression and you look at the standard expression of a neural network, there is an equivalence, but this here is much easier to interpret because we understand the meaning of placing the hidden units and changing the spread. All right, so our contribution builds on top of this, and let me just give you a high idea of what we made here. So this is a work that we submitted last week to Uncertainty in Artificial Intelligence. So here what we do is we propose an expressive prior for neural networks, and we show desirable properties with closed form analytical derivations. So we show that this model is able to encode the stationarity, as well as it allows for easy user specification of length scale and amplitude variance in a decoupled fashion. Then we also derive, so we demonstrate, we show a consistency theorem in a frequentist way, meaning that the regression function that we are able to learn will converge to the true function as we gather more and more data, as the number of observations goes to infinity. And finally, we demonstrate the successful behavior of this model empirically. Yeah, so very high level, just the key ingredients of our contribution is to have effective priors, expressive priors over these two key parameters of our model, the centers and the scales. And the idea is that we are allowing the centers to move to any location wherever it is needed. So we are controlling for the location of the centers, and then we are assuming a prior over the scales. So we're controlling the width of these activation units such that we get the two properties, the amplitude variance and the length scale decouple. Yeah, I'm happy to go into this more later in the questions, but just let me, very briefly, so like we have in the paper all these properties detail, but let me just show you, okay, what did we get with our pictures? So this is here, I'm showing samples from the prior of our functions, okay? And the Gaussian process here, so when we go from first row to the second, what we're doing is we're increasing the wiggliness of the functions. That's very easy to do with a GP. And from the first row to the third, what we're doing is by keeping the wiggliness the same, we are increasing the amplitude variance. Now notice, this is our model, the first row, the first column. Our model is basically indistinguishable from a GP. but still is a neural network. So this is the first work that actually is able to recover behaviors like a GP while being a neural network. And notice, remember a standard Bayesian neural network. This is the BNN, and we see how we have non-stationarity around the origin even before seeing data. Yeah. So we are able to get this, and then finally we can see how that can potentially help whenever we give our predictions in real applications. Here, what we get, we have the posteriors learned by two models. This is our model, and this is a standard Bayesian neural network. And both models have been tuned to have the same a priori amplitude variance and length scale. And still, as you can see, the standard Bayesian neural networks get these spurious uncertainties. It is very uncertain in certain regions where it actually doesn't make any sense to be that uncertain. So our model is much better behaved. And yeah, the underlying reason for that is that we're able to learn automatically where the hidden units have to go in order to accommodate a smooth function behavior. So all this, I find all this very important. It's about understanding what our model assumptions are and what the implications are whenever we are actually giving predictions with uncertainties. And potentially, I think this has tremendous implications for any application where you have to actually do functional predictions. Okay, let me jump very quickly into wrapping up, so in my conclusions. So this is my contributions again. Now, what's next? So I'm very much excited about bringing my research to the real world. And one current effort I'm pursuing is bringing our models in antidepressant prescription recommendations into the clinic. So for that, I'm working with the human-computer interaction experts and trying to bring the models to the Massachusetts General Hospital. Using user studies, we look at how that impacts their clinical practice and see if those systems are useful, if they are trustful, etc. And more generally, regarding my current and future research agenda, so I want my research to focus on impactful real-world problems. And so I'm working on the personalizing mental health care. I'm also very excited about starting collaboration with folks in vitro fertilization. There it's actually great because the outcome is very clear in the sense that you know if the outcome is pregnant or not pregnant. So in mental health care, it's very hard to actually even tackle the question of outcome, just because what does it mean to, we don't have labels, right, in the data. So we have the electronic health record. We have to think about what's the actual outcome of interest. But here is we have that data, and that's very exciting to develop also useful methodology. So generally speaking, I want to develop useful machine learning methodology to better assess error, to model uncertainty, and then also to be able to combine expert knowledge into the data-driven, so the machine learning systems. Finally, how to learn task-meaningful representations. Yeah, so this is the high-level picture, and I will be very happy to take questions from here. Again, I would like to acknowledge my collaborators. So thank you for listening. Yeah.
So we are running slightly over time, so whoever actually can leave now, but whoever wants to stay can ask questions.
Yeah. Yes.
So in the second half of your talk, you described this alternative to a Bayesian neural network. Can you just talk about the training of such a network? So what that looks like, what are the dynamics of it, how does it compare to a Bayesian neural network or even a vanilla neural network that isn't Bayesian?
Okay, yeah, awesome. So this work, in this work, we use Hamiltonian Monte Carlo to train our neural networks. So this is the gold standard to whenever you are interested in model properties. So this is, I would say that this work is a first step towards having more expressive and meaningful models and priors. Yeah, so Hamiltonian Monte Carlo is not super fast, right? So it is actually, the next step would be to develop variational inference or other more scalable strategies. And yeah, so we have a student that will catch up there and then continue with that. Yeah, so that's.
All right. Thank you.
Thank you so much.